Since an early age, I've always been fascinated by algorithms. There was this game called P-Robots that I loved when I was a kid. It was a programming game in which you have to code your strategy for a robot to fight other robots and hopefully beat everybody else in the competition. Over time, I managed to code stronger and stronger robots. In a way, that's exactly like how evolution works. That's what organisms do when they adapt to an environment and compete with each other. In a way, I'm combining both passions now as an evolutionary geneticist. I want to track how species adapt and evolve, and we do it with both top-down and bottom-up approaches. We track populations as they evolve, and we also generate F1 hybrid stem cells between mouse species and manipulate them to basically put genes into the uncomfortable position of having to work with their long-lost breath within a new cell. It's fun trying to understand how the genome works. Next-generation sequencing is central to our work. We don't go a day without talking about it in one way or another in the lab. It's basically the most powerful molecular microscope you can have. In my lab, we regularly use all kinds of sequencing, from single-cell transcriptome or chromatin profiling to whole genome sequencing in thousands of individuals. The kind of granular detail and insight you can gain into a system is just simply unprecedented. In a way, the through line in our work is to have a quantitative understanding of what's going on inside the body and inside the cell. And in today's day and age, there's no other tool that's as sharp and as fine-tuned as next-generation sequencing. I still remember way back then when I was still a grad student. It's been like 12, 13 years ago now, when we first got the genome analyzer tool in our lab. Back then we still call it Solexa. You have this ginormous machine, right? And then there's this tiny window there. You reach in, you have to grab the prism, and then you have to clean it. You have to polish it, and then use a pipette, reach in and add some mineral oil on top to make sure that the imaging can happen. It was a chore, but it was so exciting to see how this whole thing works. Around the same time, we're designing the first SNP genotyping array for the sticker packs. It wasn't a very good array, but we still managed to get many papers with it, and it was so transformative. And so you just know that if people can sequence cheaply and easily, how powerful that would be. In a way, since we started our own lab, we started off mainly as users, but then you just can't help yourself getting excited by wanting to get more and better data if only you tweak the technique here and there. And the thing is, I still remember the tantalizing feeling when you feel like you can just touch and understand and grasp something and understand an organism by sequencing the genomes. So with the techniques we develop, we want to enable other people as well so that they can share in that sense of possibility. Well, with haplotype resolved sequencing, really we're talking about linkage. Linkage is essential because linkage and haplotype are the glue that makes everything work in genetics. Just think about it. Right now we're living in the middle of a pandemic. We talk about tracing and tracking the B117 variant, but not as two separate E484K and the N501 spike protein mutations, because the whole thing's got to work together. And nobody just goes in there and types those two mutations. Instead, they sequence the whole thing. In the same way, in eukaryotic diploid genetics, regardless of whether you explicitly acknowledge it or not, you're working with linkage. Let's take genetic mapping. Most any mutations we are tracking in mapping are themselves neutral. Only really rarely would you be directly typing the causal mutation itself. And when we talk about mapping specific variants in GWAS, for example, often when people dig deeper into them, they find interactions either between mutations or with the genetic background. So in a way, with the way the sequencing field is evolving, you really need to start thinking about how you can describe the genome in the quickest, best possible way. The game is no longer just lowering the per base cost. Rather, it's about doing it well. So haplotype resolved sequencing, that's where the field is going, whether you like it or not. Either it's with linked read sequencing like haplotagging, or long read sequencing. In fact, with everything else that's been sequenced out there, I argue that until we get to the point where we can read the chromosome from end to end perfectly, linked read sequencing has a very important role to play. That's because A, it is scalable, B, it's accurate, and ultimately C, for large populations, you don't really need to see everything to be able to call genotypes perfectly. Just think about what you have in your pocket, your phone, right? There's the predictive typing feature for your phone and email. How many times are you surprised that actually your phone does a better job of finishing your sentence than you do? In the same way, with everything out there being sequenced, why resequence absolutely every single base when instead you can just skim along the chromosome and directly haplotype like what we have shown in the Davies et al. Nature Genetics quote paper. That is going to give you your best value for your sequencing model. So yeah, haplotype resolved sequencing is absolutely the future. And I'm of the opinion that thus far linked sequencing basically hasn't exactly been pitched right. It's going after wrong applications like building de novo assemblies or phasing in single individuals. In my mind, its strength is really in large population typing. And if that's correct, then short read sequencing empowered by linked sequencing is going to be the workhorse for all kinds of genetics for a long while to come.